welcome to a voiceover of the video that I did in regards to the cell membrane and protein function. I think this first little interlude isn't that important, so I'm going to skip a little bit ahead. So right now I'm drawing what's known as the phospholipid bilayer, which is the cell membrane. This again is the fatty acid structure that we created before break. We could also call this the plasma membrane. So cell membrane, plasma membrane, phospholipid bilayer, they're all the same. And that's because we have that double layer of phospholipids. Those circles, the heads, are polar, and the tails are non-polar, and they could be saturated or unsaturated fatty acids. Now, within that plasma membrane, as I'm going to draw in a second, there are a lot of proteins that are embedded, almost like gates or doors in a castle wall, that allow the cell to receive a lot of information. So that green square right there is what we call a receptor protein, and that's going to be designed and created by the cell to receive information from the outside world. The bloodstream, other cells, other tissues, could be a lot of different sources that go into it. And again, this is outside, so probably that outside space would be what we would say is the bloodstream. The bloodstream is your body's best way of transporting different things throughout the body to communicate between different cells. So at the edge of this receptor protein, what I'm drawing in red right now is the active site. The active site is going to be the place in which a signal molecule or a hormone can bind to. Think of it in an analogy as when you go home and you unlock your door, that's the location in the door that you put your key into. It's really specific in terms of a three-dimensional shape based on amino acid sequences that are going to allow a signal molecule to bind to it. Most cells have hundreds of different receptor proteins, each designed to bind to a very specific signal molecule. And signal molecules tend to be very small proteins that can flow throughout the body. Those blue lines that I just drew there are hydrogen bonds that are temporary interactions between the receptor protein and the signal molecule, which means there's going to be positive and negative interactions between both sides. This is not a permanent connection, just like you don't leave your key in the door. This is merely just a way to make the receptor protein know that something is happening in the body and we need to turn it on. I believe you took notes earlier this week about different types of hormones. Insulin and glucagon are two hormones your body makes to help regulate blood sugar. Many of you might be familiar with melatonin, which is a hormone that helps regulate your sleep cycle. Serotonin is another neurotransmitter hormone that's helpful in regulating mood. Dopamine obviously is useful in creating those feel-good those feel-good emotions. There are so many different types of hormones in our body, each of which has a specific type of receptor protein. That receptor protein is going to change its shape as a result of that signal molecule binding to it, almost like a key going into a lock can unlock the door. And that change in that protein shape is going to result with changes happening in the cell. More on that next week. The big thing that happens, though, is some cellular response is going to occur. And that cellular response, that final change within the cell, is probably going to be targeting what we will soon draw as the nucleus. So right now I'm drawing the nucleus. The nucleus is often called the command center of the cell. It is double membrane bound, which means it has an inner and an outer layer of phospholipids. And that's really designed to just help protect the genetic information of the cell from damage. Again, we call this the nucleus. And the nucleus, as I will draw it in a second, has small little pores called nuclear pores that allow things in and out of the cell. They're tiny little tunnels that are very specific to letting things in and out. Sort of like the front door at our school. It's really the only place you can get in and out of the school during school hours. Often as a result of a cellular response due to a signal molecule, that tiny little red dot known as a transcription factor, I'm going to write that out in a second, can flow into the nucleus, and it will find a region of DNA to help express a trait, and that trait will almost always be a protein. DNA is a molecule with two sides, and it's made up of the nucleotides, which we learned about before break. One is adenine, guanine, cytosine, and thymine, AGCT, and DNA is just a repeating sequence of those four letters. A transcription factor finds that region of DNA, binds to it, and helps create a copy of that DNA. So the cell is able to use that information to make a protein. Sort of like opening a recipe book. You can open a recipe book at any point or time and find a recipe to make mashed potatoes or chicken fingers or grilled cheese or tomato soup or lasagna, and you can then go and make that recipe. But that recipe contains that copy of that information all the time. So out of the pore is going to flow a small band of genetic information. This genetic information is only single-sided, and it's known as RNA. We're right now just talking about these ideas in big, broad terms, but we're going to spend multiple weeks in our fourth unit in genetics talking about these types of molecules and these processes. But RNA is the temporary copy of information that's used to create our protein outside of the nucleus in a big organelle called the endoplasmic reticulum. And like the nucleus, it is embedded within another phospholipid bilayer, which is why I've drawn that black line. We're going to be making those proteins inside of the endoplasmic reticulum using a small structure known as a ribosome. And that ribosome, almost like a pair of eyes, reads over the RNA. Oops. It reads over the RNA like a pair of eyes to make the protein. And it's going to be just adding amino acid after amino acid as a result of reading the RNA. Sort of like all of you rolled the die 20 times to make a protein using, different types of M&Ms. Really, we can think about the endoplasmic reticulum as a protein factory. It's just going to be spitting out proteins as a result of genetic expression. That green squirrely line right there is the finished protein as a result of forming an alpha helix, which is our secondary structure, and then having all of the R groups form the tertiary structure, where we have the complex folds, bends, and curves as a result of those R groups. And once we do that, we're going to be almost done with our protein. But the endoplasmic reticulum just makes the protein. It doesn't finish it, it doesn't specialize it, it doesn't garnish it, modify it, or check it, which is why we have a second organelle that's also surrounded by a phospholipid bilayer. And this is called the Golgi apparatus. The Golgi apparatus is a series of folds, almost like a pancake, a stack of pancakes. And the Golgi complex, or the Golgi apparatus, has each of those stacks surrounded by a phospholipid membrane. This is going to be where we modify and specialize proteins. We're going to be specializing and modifying them by adding small little attachments, bullying, you might say, to the protein that we just created in the endoplasmic reticulum. We, however, need to be able to get there. So we're going to use a ROAD, a protein known as a microtubule, to get us to that space. The microtubule is going to be a thin strand of protein that the body makes. This is a structural protein, so it's repeating filament. We're going to be bringing that protein in a small little bubble called a transport vesicle. And the thing bringing it over is another type of protein that almost walks like an AT-ST walker in Return of the Jedi, if you know your Star Wars. And if you don't, think about a pair of legs just walking very slowly across the microtubule. And it's going to be carrying it almost like a giant balloon behind its back. And these motor proteins are a type of structural protein, but also a type of enzymatic protein because they can react with energy to do this work. So I'm sort of showing right now in the dark room how it might move across. Probably didn't do it justice, but we'll watch a video of how that process works next week. That little vesicle is going to fuse on the cis side of the Golgi complex, and each of those numbers that I'm writing represents sort of like a finishing station where we might be adding a very specific molecule or modifying its structure ever so slightly to make it a specific protein. And when it's finished, it will fuse out of the trans side in another transport vesicle on another microtubule using another motor protein. And its final destination, we'll talk about in a minute, will either be being embedded in the cell membrane or being spit out into the external environment. Right now I'm drawing the bling. The bling are the specific additions to the protein structure that make this protein a finished product. In a way, well, the two things that we can add are often lipids or carbohydrates in small amounts. So either CHs or COHs. And in a way, it's sort of like going to a hardware store and you get a master key, and then you have the keysmith or the key tool notch in the really specific edges that make that house key become your house key, or that Subaru key become your Subaru car key. That way no one can just go and unlock your house or unlock your car and drive away with it. We need those specific little additions to make that protein really, really specific. This process of putting it out of the cell and into the bloodstream is called exocytosis. Exocytosis is going to be the process where we spit out that protein. I think I'm now doing a grand overview, so let's go back over it. Signal molecule binds to a receptor protein as a result of hydrogen bonds between amino acids. That's going to result in a transcription factor, another protein going in and telling DNA to be copied. We're expressing a gene that will result in a single copy of RNA going into the endoplasmic reticulum and being copied by a ribosome. That ribosome looks or reads the RNA to make the protein, which folds into the tertiary structure. We use a transport vesicle using a microtubule to bring the protein to the Golgi complex, where it fuses on the cis side. We modify and add things to it. It exits the trans side, and then finally we're ready to put it in our cell membrane or to put it out in the bloodstream to do more work. And that is the general process of what we call the endomembrane system, which is how we make proteins in all of our cells.